Hello, welcome to another screencast where we'll be finishing the genetic variation discussion. So this is a continuation from genetic variation 1. So we're just picking up here in the middle. And what we're trying to do here is to focus down here on C this time. So some sources of genetic variation at the chromosome level, talking about meiotic errors, that is non-disjunction, some chromosomal translocations, and then this polyploidy stuff that happens in plants, which is not really common in animals. Okay, so so at the chromosome level, not initially at the individual, gene level, DNA level, we can have chromosomal level alterations can produce genetic variation. Right? And so if we see here in A deletion removes a chromosomal segment, right? So if this segment, this segment D, right, after we delete it, then we just have this long arm being a little bit shorter. So this does happen. A duplication that repeats a segment, right? And so here is this BC, and so we duplicate that, and then we go like BC, BC, and we make that longer arm even longer. Right? So this may be sounding like a mutation, but a mutation is more at like the point level, more at like the actual DNA nucleotide sequence level. Here you're talking about, long stretches of sequences that are being either deleted or duplicated. Another way is to invert it, right? And so here's our sequence BCD. These are segments BCD, and then you invert that and you go DCB. So this changes the reading, right, of it there. And then finally a translocation, which is just what it sounds like. It's you're changing locations, right? Moves a segment from one chromosome to another non-homologous one, right? So this is not crossing over, right? This is not a recombination between homologs. This is between non-homologs, okay? And so here's a longer chromosome, here's a shorter chromosome, and we get this reciprocal translocation, meaning that this AB segment and this MNO are going to be switched. So now on this longer chromosome you have MNO and you have AB where you didn't down here. So you're generating recombinant chromosomes there, but not homologously. Right? So an example, a famous example of this chromosomal translocation deals with chronic myelogenous leukemia, or CML, and you end up getting this, quote, Philadelphia chromosome here. So this happens when chromosome number 9 and chromosome number 22, you exchange, these segments right here, okay? So this is the tip here and the tip here are exchanged. So you end up getting, this is the Philadelphia chromosome now, which is shorter, right? So you removed this part here, and you removed this part from 9, and this part from 9 gets added to up there. So this is not reciprocal. This is the, it is reciprocal, because then this ends, up on the longer chromosome there, right? And again, so this is in some way linked to this chronic myelogenous leukemia, a type of leukemia there, and this is just another diagram of the same thing, right? So in meiosis, you can have not just like those are involving segments of the chromosome. Here in meiosis, you get complete chromosome non-disjunction. So this is another way at sort of at the chromosome level, right? So this would be a 2. So this is the second way here, and you get this non-disjunction where, for some reason, at metaphase at the metaphase 1 plate, right, and in anaphase 1, you did not get the separation of this tetrad. You did not get this homologous chromosome separating. These ones separated normally up here, right, but this pair did not. And so then the result is, again, 2 cells here. This cell is missing a chromosome, so that's -1, and then this one is up a chromosome, right, so this is going to be +1. Right? So you still have, then in meiosis 2, this is going to be meiosis 2 with the chromatids separate, and then that's how you end up getting these haploid cells, but it's haploid plus this extra chromosome right there, and again, this one's going to be haploid plus this extra chromosome right there, and then these get haploid, but they're down a chromosome, and haploid, but down a chromosome here because of this non-disjunction at meiosis 1 at anaphase 1. Another spot of non-disjunction would be down here at meiosis 2 or at anaphase 2. Right? So at anaphase 2, you're going to get normal, disjunction here. You get normal, so this is going to be an N cell. This is going to be an N cell, haploid cells. You get normal separation of all the homologous chromosomes here, so everything's fine and dandy. But then, for some reason here, you don't get the equal splitting of the sister chromatids, and so this is going to yield you N+1 cells, N-1 cells over here because of this non-disjunction. But over here on this cell, this was normal, right? You got this normal homologous chromosome over here, and then you got normal regular separating in anaphase 2 of the sister chromatids over there, and so you end up generating just these normal cells over there. So in order to know where the non-disjunction occurred, you have to ask the question, okay, what gametes or what haploid cells did you get? If you got N+1s and N-1s, then the non-disjunction happened up here in anaphase 1. If you got N+1s, N-1s, and regular N cells, then the non-disjunction most likely happened in anaphase 2. Right? So there's a little animation here to sort of watch that happening. Take a look at that. The famous examples of these non-disjunctions are after fertilization. So you get non-disjunctions, but then you get normal regular fertilizations, right? And the result could be trisomy 21 or Down syndrome. So trisomy, this term trisomy, right, means that you have 3 chromosomes, right? Somy, 3 individual chromosomes, of which one, in this case, it's number 21. Right? So if you look at the karyotype here, there are 3 chromosome number 21s, and to understand this, right, how would you get this, right? You should be able to describe and explain how you would end up getting this trisomy 1 situation, meaning the gamete, this gamete, and this gamete, when they came together, right, how did you end up getting the karyotype of this? So what happened here? What gamete would this be, and what would the gamete be there? What kind of chromosomes would you have there? Right? Klinefelter syndrome is another one resulting from the non-disjunction, this time of an X chromosome. So this is non-disjunction of chromosome number 21, right? And this would be of a sex chromosome. Right? So in this case, the trisomy is that you have 3 of the sex chromosomes here. You get 2 X's and a Y. And to show an understanding, you should be able to say, okay, which gametes fused together, right, to yield this situation right there. Right? There's another situation not shown, but this is Turner's syndrome. And people that are Turner syndrome are X0, right? That means that they only have 1 of the sex chromosomes. Here they have XXY, so these people are XXY. But Turner's is X, and then there's nothing else over there. So again, to be able to demonstrate that you understand what's happening there, you should be able to describe what the gametes would look like to get that Turner syndrome. Okay? Finally, polyploidy. Polyploidy is something that happens in plants, and this is different than polysomy, right? Polyploidy refers to an entire set of chromosomes. So in a trisomy, you end up getting an extra 1 of the chromosomes, right? Here in a triploid or a triploid situation, you'd end up getting 3 whole sets of this. And this is something that happens in plants apparently pretty regularly. Right? And plants can sort of tolerate this, right? So it all goes back to, again, meiotic errors here. So errors in meiosis. Right? So follow these diagrams through to see how you end up getting this tetraploid situation, right? Where you now get 4 ends, right? And this situation exists in plants, right? And then another variation on this polyploidy, right, is down here, and I'm not going to step through that at the moment, but polyploidy getting a whole extra set of the chromosomes, right? How might that happen? How might disjunctions there, non-disjunctions happen to generate that, right? So combining with the genetic variation 1, this is a quick sort of overview then of sources of variation at the chromosome level, right? There's non-disjunctions, chromosomal translocations, and then polyploidy in plants. Right? And again, the take-home message from all of these is that these are all mechanisms, these are all ways, these are all sources of generating genetic or DNA sequence variation within populations. Right? So in class we'll also discuss a little bit more specific there about meiosis and random fertilization, random mating, etc. Right? But these are all sources of genetic variation. Okay, thanks for listening. Bring your questions to class.